Hello. Good evening to the students who have joined. Can you please let me know if my voice is audible and is my video is visible? Let me know. Okay. Thank you for confirming. Please let me know if you can see the presentation. Is the PowerPoint presentation visible to you? Okay. We will wait till it's 6 p.m. Then we will start the session. Okay. Meanwhile, if you have any doubt or any query regarding our last class, whatever we have discussed, feel free to let me know. We can discuss those things right now. 5 more minutes, then we will start the session. Okay, so kindly wait. Okay, so it's almost the time. I think I should start. Okay, so it's time. Now let me start the session. Hmm. Let me share the PPT. So I hope my voice is still audible and you can see the presentation clearly. So very good evening to the students who have joined. My name is Mousumi Deb and I'm a PMRF fellow from the Department of Chemistry and Chemical Biology, IIT ISM Dhanbad. I'm your TA for this NPTEL SWAM course of Medicinal Chemistry. Today it is our week 6 live session. So before starting the session, like every week, I would like to summarize a few things that these problem solving sessions are scheduled on every Monday from 6 p.m. to 8 p.m. and it will be for it is a, since it is a 12-week class, we will have a 12 problem solving sessions each week along with a summary session that will be on at the end of the 12-week course. Okay, we will have a 1 or 2 hour long summary session where I will summarize the entire course. So these sessions are live on Google Meet platform and this is the link which will remain same throughout our sessions. Okay, you can write it down anywhere or you can save it and use it for joining each Monday class. If any network disruption happen from my side, do not leave the session. I will try to rejoin as soon as possible. And during the session it is open. The call is Google Meet call is open. So if you have any problem with your network, you can leave and then you can rejoin at any time without asking permissions. Okay. So, since I'm recording the session, I am muted your microphones and cameras so that I can record the session without any problem. Okay, but the chat box is always open. So, if you have any doubt, any query, or anything you want to tell me, you can let me know through the chat box. Okay, I am recording the session, and this recording and the PPT which I am sharing today in the class both will be shared to you via the SWAM portal. Where to find it, I will show you next. Okay, so okay, important thing is during the session we will use the chat box for the question answer session and as well as we will use the chat box for any kind of doubt or any kind or everything. Okay, so if you have any problem you can let me know through the chat box. Otherwise, if after the session you have still you have the queries, you can let me know via email. I will share my email ID at the end of the session. So first let me show you where to find these materials. I have shown you previously also, but let me do it one more time. So first you have to go to this SWAM portal. Okay, on the announcement page. Okay, in the announcement page you will have all the course announcement. On the left panel, as you can see, there is a weekly course. There is a weekly course update. Okay, so on the left side you can see there is a weekly course update. Here there is a problem solving session is written, right? If you click on that point and go to problem solving session recording, you will be redirected to a page which looks like this. It will have each and every date, each and every YouTube video link for the previous class recordings. Also, it contain a presentation file which is a Google Drive folder which contains all the presentation whatever we have I have done so far. Okay, so since we have done already around 5 live sessions and today it is the sixth one, so here already we have the 5 PPTs and the sixth one I will upload it after today's session. Okay, so these things keep it in mind. So where to find the sessions and all the things. Okay, so in the power problem solving session, how we do it? I will show you around 20 questions from the week 6 course content. Okay, and each question will have 4 different options. After showing a question, I will wait for 1 to 2 minutes, and after that I will check the chat box. Whatever you think is the correct answer, you have to let me know in the chat box. Okay, for answering in the in the class there is no negative marking, so you can answer in a free mind. Okay, but the quizzes whatever is posted on the NPTEL SWAM portal, they are graded. Okay, so be careful while answering the assignment portion. Okay, so each and every question will have a descriptive answer which will help you to write the descriptive questions regarding the same topic and understand the course better. Okay, So let me start the session. So what is the topic of the week 6 course content? So the topics are the pharmacokinetics part 1, part 2 and drug metabolism part 1, 2, 3 and 4. So basically today the main two topic which we are going to discuss, one is pharmacokinetics and other is drug metabolism. So now when we will go into the detail you will understand, and there is ADME also. ADME I will tell you later what is ADME. So whatever, so basically pharmacokinetics, the drug metabolism comes under the pharmacokinetics. Okay, it is a topic of pharmacokinetics itself. So in the last class, as I have remember, we have discussed about the nucleic acids as the drug target. We have discussed various nucleic acid based drug targets like chain cutters and chain terminators, topoisomerase, poison inhibitor, and so on. And also we have discussed about a little bit about antigen, antibody, hapten, enzymes, the methods of biological chemistry, and a little bit about the metabolites also. From now on, we will be getting into more detail into the mechanisms which are involved in the medicinal chemistry, and the start is from today, that is the pharmacokinetics portion. Okay, so let me start with the first question of today's session. So the first question, it says, which of the following statements best describes the pharmacokinetics? Okay, your options are as follows. Number A, the study of how drugs reach their target in the body and how the levels of a drug in the blood are affected by various factors. Number B, the study of how drugs can be designed using molecular modeling based on drugs pharmacophore. Number C, the study of how the drug interacts with its target binding site at the molecular level. And number D, the study of which functional groups are important in binding a drug to its target site and the identification of pharmacophore. So, which of the following statement best describes the pharmacokinetics? Again, I will repeat. Number A, the study of how drugs reach their target in the body and how the levels of the drug in the blood are affected by various factors. Number B, the study of how the drugs can be designed using molecular modeling based on the drug's pharmacophores. Number C, the study of how a drug interacts with its target binding site at the molecular level. And number D, the study of which functional groups are important in binding a drug to its target site and identification of pharmacophore. Which of the following statement best describes the pharmacokinetics? I will wait for a minute or two and then I will check the chat box. Okay, read the options carefully and let me know A, B, C or D, which of the following you think is the correct answer for problem number 1. Which statement best describes the word pharmacokinetics? After checking the answers, I will discuss what is pharmacokinetics, what is its importance in medicinal chemistry and also overall in our body. Which of the following statements best describes pharmacokinetics? Let me check the chat box. I hope to find some answers there. Okay. I have received two answers and both are saying that number A is the correct answer. Okay, now let me check if number A is the correct answer or not. Yes, the correct answer is number A, the study of how the drugs reach their target in the body and how the levels of the drugs in the blood are affected by various factors. So basically, pharmacokinetics, or in short, it is known as the PK, is the study of what the body does to a drug from the moment of administration until it is completely eliminated. It focuses on the time course of the drug concentrations in the biological fluids and tissues, which is essential for determining safe and effective dosing. The ADME framework, the core of pharmacokinetics, is categorized into four main processes, commonly abbreviated as ADME. And previously, when I was discussing the contents of today's class, there was a term ADME which I had not discussed that right time. So the ADME, it is a part of the pharmacokinetics. It basically, this is the framework of the pharmacokinetics. Okay. So what is the ADME? So A stands for absorption. Okay, the process by which the drug enters the systematic circulation from its sites of administration. For example, like GI tract or skin. And in the administration, we have this term bioavailability, the fraction of the administered dose that reaches to the bloodstream unchanged. IV, for intravenous drug, the bioavailability is 100%. Okay. Distribution, D stands for distribution, the reversible movement of the drug throughout the various body fluids and tissues. And the parameter which is important for distribution is the volume of distribution, or VD, the theoretical volume related to the amount of drug in the body to its concentration into the plasma. It indicates whether a drug remains in the blood or dispersed into the tissues like fat or muscle. Then we have the metabolism or biotransformation, the chemical alteration of the drug primarily by enzymes in the liver. Then we have phase 1 and phase 2 metabolism. These reactions usually convert the lipophilic drugs into more water-soluble metabolites for easy excretion. And excretion, the irreversible removal of drug and its metabolite from the body primarily through kidney or via bile, feces, lung and sweat. Okay, this is the ADME framework and the pharmacokinetics. So basically, pharmacokinetic, in a easiest term, it can be described by what the body does to a drug from the moment of administration. Another term which will be important and that we will discuss later on is the pharmacodynamics. So together, pharmacokinetics and pharmacodynamics are known as the PKPD parameters. Okay. So, this pharmacokinetics is what the body does to a drug, which is the absorption, distribution, metabolism, and excretion. And pharmacodynamics is what that particular drug is doing to our body. Okay, for example, like it is having the microbacterial effect, it is showing the inhibition or killing to some bacteria, or it is showing some inhibition to some enzymes or some adverse effect. Okay, so these all the effects are discussed in the as the pharmacodynamics. Okay, so this is the two very important term in our medicinal chemistry, which is the pharmacokinetics and pharmacodynamics. Okay, so this is so yeah, the number A was the correct answer. Now I will go to the next problem. So problem number 2, it says that which of the following drugs does not obey Lipinski's rule of 5? Your options are number A, atorvastatin, number B, rosuvastatin, number C, vinorelbine, and number D, all of the above. Which of the following drugs does not obey Lipinski's rule of 5? Atorvastatin, rosuvastatin, vinorelbine, and all of the above. Again, I will be waiting for 2 minutes and then I will check the chat box. Let me know A, B, C, or D, which of the following is the correct answer. Which of the following drugs does not obey Lipinski's rule of 5? Atorvastatin, rosuvastatin, vinorelbine, or all of the above? Which of the following drugs does not obey Lipinski's rule of five? Let me check the chat box. Let's see if I have any answer in the chat box. Okay, still no answer. I have expected one or two answers for the problem number two. Okay, let me show you what is the correct answer for problem number two, then let us discuss it. Okay. So the correct answer, the drug which that does not follow the Lipinski's rule of five, the correct answer will be, it is number D. All of the three drugs, atorvastatin, rovastatin, and vinorelbine, all these drugs does not obey the Lipinski's rule of five. Now, why these drugs does not follow Lipinski's rule of five? Already I have discussed what is Lipinski's rule of five, right? We all know that the molecular weight of the drug should be less than 500 Dalton, and then we have this so the rule of five has been extremely useful rule for thumb for many years, but it is neither quantitative nor foolproof. For example, orally active drugs such as the atorvastatin, rosuvastatin, cyclosporin, vinorelbine do not obey this rule of five. It has also been demonstrated that the high molecular weight does not itself cause poor oral bioavailability. One of the reasons that the molecular weight appears to be important is that the larger molecules are invariably have too many functional groups that is capable of forming hydrogen bond. So basically, in case of the Lipinski's rule of five, it has been stated that the drugs which does not follow the Lipinski's rule of five, they are not they are not very good, or for a orally active for a drug to be orally active, or a drug to be able to taken orally, the administration, so it must follow the Lipinski's rule of five. Okay, so what is the rule of five? I think I have discussed it in our first class itself, that the rule of five encompasses of mainly four different rules, and that the number of hydrogen bond donor, number of hydrogen bond acceptor, the log p value, and molecular weight value. So these are the parameters. Okay, so in case that particular molecular weight, it has been stated that the drugs which have a high molecular weight, they does not they cause poor oral bioavailability. But the exceptions are there. For example, atorvastatin, rosuvastatin, vinorelbine, all these drugs, they are very good orally active drugs, but they does not follow the Lipinski's rule of five, because as you can see, their structures are huge and their molecular weight is greater than 500 Dalton. Okay, so that is why these drugs, but still they are the orally bioactive. So that is why this rule of five, Lipinski's rule of five, it is neither it is not foolproof. Okay. So the correct answer was number D. All of the above, all of the drugs, they does not obey the Lipinski's rule of five. Now I will go to the next problem. So problem number three, it says that most of the current drugs are metabolized by dash primary CYP enzymes. Fill in the blanks. Your options are number A 2, number B 7, number C 4, and number D 5. Most of the current drugs are metabolized by uh, dash number of primary CYP enzymes. You have to fill in the blanks. Your options are 2, 7, 4, and 5. Let me know what will be the correct answer for problem number 3. Most of the current drugs are metabolized by which number of primary CYP enzymes? most of the current drugs are metabolized by, what will be the correct answer? Let me check. Okay, I have received a couple of answers. So, number B, number C, number B, number D. Okay, so I have received number B, C, and number D as the answers. Okay, now let me check which of the following will be the correct answer for our problem number 3. So the correct answer will be, it is number D, 5. So total by 5 CYP enzymes, the most of the current drugs are metabolized. Okay. So this is the relative importance of the CYP 450 drug in the metabolism. So we have this, these are the 5 most important CYP enzymes: CYP 3A, CYP 2D6, CYP 2C, CYP 1A2, and CYP 2E1. So there are at least 33 different cytochrome P450 or CYP enzymes grouped into four main families for CYP1 to CYP4. Within each family, there are various subfamily designed by a letter, and each enzyme within that subfamily is designated by the number. For example, CYP3A4 is the enzyme 4 in the subfamily A of the main enzyme family 3. The most drugs in the current use of the metabolized by 5 primary CYP enzymes. So what are these 5 primary CYP enzymes? These are CYP3A, CYP2D6, CYP2C9, CYP1A2, and CYP2E1. The isozyme CYP3A4 is particularly the most important in the drug metabolism and is responsible for metabolism of the most of the drugs. So this is very important. Remember that the isoenzyme CYP3A4. So this is very important. Okay, it's this CYP3A4 is responsible for the metabolism of the most of the drugs. Okay. So the correct answer was number 5. These are the families of the CYP enzymes which are most crucial for the drug metabolism, and isozyme CYP3A4 is particularly most important for the drug metabolism. Okay. So now I will go to the next problem. Problem number 4. Which of the following equations is correct for the Henderson-Hasselbalch equation for the weak bases? Your options are: number A, pH equal to pK minus log of unionized drug concentration by ionized drug concentration. Number B, pH equal to pK minus log of ionized drug concentration divided by unionized drug concentration. Number C, pH equal to pK plus log of unionized drug concentration by ionized drug concentration. And number D, pH equal to pK plus log of ionized drug concentration divided by unionized drug concentration. Which of the following equations is correct for the Henderson-Hasselbalch equation for the weak bases? Take this into account, that what will be the correct answer, what is the correct form of the Henderson-Hasselbalch equation for the weak bases. Again, I will wait for 2 minutes and I will check the chat box. Let me know A, B, C or D, which of the following is the correct answer for problem number 4. Which of the following equations is correct for the Henderson-Hasselbalch equation for weak bases? pH equal to pK minus log of unionized drug concentration by ionized drug concentration. Number B, pH equal to pK minus log of ionized drug concentration divided by unionized drug concentration. Number C, pH equal to pK plus log of unionized drug concentration divided by ionized drug concentration. And number D, pH equal to pK plus log of ionized drug concentration divided by unionized drug concentration. For another minute I will wait, then I will check the chat box. Okay, let me check if there is any answer for problem number 4. Number D, number C, number D, number D. So I have received three number D and one number C as the answer. Okay, let me check what will be the correct answer, number D or number C. So the correct answer is, it is number C. Okay, so pH equal to pKa plus log of unionized drug concentration divided by ionized drug concentration. So the extent of ionization at a particular pH can be determined by the Henderson-Hasselbalch equation, which is this one. Okay, see, it is the unionized drug concentration divided by ionized drug concentration. So this is this form. So for weak bases, this is the equation, pH equal to pK plus log of RNH2 divided by RNH3 plus. This is the unionized, this is the ionized form, where the RNH2 is the concentration of the free base and RNH3 is the concentration of ionized amine. K is the equilibrium constant for the equilibrium shown in the figure. So this is the equilibrium. So this is the ionized amine, as you can see. This is the ionized amine NH3 plus. So this is the receptor interaction and water solubility, and then after crossing the membrane, unionized form, which is the free base, equilibrium between the ionized and unionized form. So K is the equilibrium constant for this particular equation. Okay, and so the this now this Henderson-Hasselbalch equation can be derived from the equivalent equilibrium constant. So K will be the product of the concentration of H plus and RNH2 divided by the concentration of RNH3 plus. Okay, so therefore pK equal to minus log H plus into RNH2 divided by RNH3 plus. So minus log H plus minus log RNH2 divided by RNH3 plus. So now pH equal to minus pH equal to minus log RNH2 divided by RNH3 plus. So pH equal to pK plus log of RNH2 by RNH3 plus. Note that when the concentration of the ionized and unionized amines are identical, means when RNH2 equal to RNH3 plus, the ratio will be 1. So log 1 equal to 0. So then the Henderson-Hasselbalch equation will satisfy pH equal to pK. So in other words, the amine is 50% ionized now at the pH equal to pK. Therefore, drugs with a pK of 6 to 8 are approximately 50% ionized in the pH of 7.4 or the slightly acidic pH of the intestine. Okay, so this implication is very important. What is the requirement of understanding this equation or so on? So as you can see, at when there is a 50% ionization happens, then pH equal to pK, right? So therefore the drugs with a pK of approximately 6 to 8, they will be 50% ionized at the blood pH of 7.4 or the slightly acidic pH of the intestines. Okay, so that is this is the importance of this equation in our pharmacokinetic study. Okay. Now I will go to the next problem. Problem number 5 says the process where the drug is engulfed by the cell membrane and the membrane bound vesicle carry the drug across the cell is known as number A, osmosis, number B, diffusion, number C, pinocytosis, and number D, mitosis. Again, I will repeat, the process where the drug is engulfed by the cell membrane and a membrane bound vesicle carry the drug across the cell. It is known as number A, osmosis, number B, diffusion, number C, pinocytosis, and number D, mitosis. Let me know A, B, C, or D, which of the following you think is the correct answer for problem number 5. The process, what does this process called? Where the drug is engulfed by the cell membrane and a membrane bound vesicle carry the drug across the cell. Osmosis, diffusion, pinocytosis, mitosis. What does this process called? When the drug is engulfed by the cell membrane and a membrane-bound vesicle carry the drug across the cell. Is it osmosis? Is it diffusion? Is it pinocytosis? Or is it mitosis? Let me check the chat box now. Okay. Number C, number C, number C. So I have received three answers and all are saying that number C is the correct answer. Okay, let's check what is the correct answer for problem number 5. So the process where the drug is engulfed by the cell membrane and a membrane-bound vesicle carry the drug across the cell is known as, very good, it is number C, pinocytosis. So occasionally, polar drugs with a high molecular weight can cross the cells of the gut wall without actually passing through the membrane. This involves the process known as pinocytosis, where the drug is engulfed by the cell membrane and the membrane-bound vesicle is pinched off to carry the drug inside the cell. So as you can see, there is a drug and there is a cell membrane, it is getting attached and a vesicle is created, which basically passes this drug throughout the cell to the out. So this vesicle then fuses with the cell membrane to release the drug on the other side of the wall. So this process is pinocytosis and is very important for the in case of the polar drugs with high molecular weight. Okay. So pinocytosis, as you can see, solutes or drugs that bind to the cell membrane like this. Then cell membrane fold itself like this. It starts to fold inward and making a vesicle. Outer side there is an extracellular fluid. Okay, and in the cell we have as usual the nucleus and the cytoplasm. Then the cell membrane continues to fold and to both ends meet and producing a vesicle which is known as pinosome. Thus the term, remember the term. Okay, this membrane-bound vesicle which carries the drug inside, it is known as the pinosome, and the method it is known as the pinocytosis. Okay. Then then as usual, as you can see, this pinosome carries the drug to the other side, and then again the fusion happens, and the drug is released to the other side of the cell. Okay. And so this is the method of the pinocytosis and how it happens. Now I will go to the next problem. So problem number 6, it says that which of the following statements is closest description of the phase 1 metabolism? Your options are number A, reactions which occur in blood supply. Number B, reactions which add a polar functional group to a drug. Number C, reactions which occur in gut wall. And number D, all of these. Which of the following statements is the closest description of the phase 1 metabolism? Number A, reactions which occur in blood supply. Number B, reactions which add a polar functional group to the drug. Number C, reactions which occur in gut wall. And number D, all of these. Let me know A, B, C or D, which of the following will be the correct answer for problem number 6. Which of the following statements is the closest description of the phase 1 metabolism? Again, I will repeat. Number A, reactions which occur in the blood supply. Number B, reactions which add a polar functional group to a drug. Number C, reactions which occur in gut wall. Number D, all of these. Let me wait for 1 to 2 minutes, then I will check the chat box. Which of the following statements is the closest description of phase 1 metabolism? Let me check the chat box. Let's see where's the answers. Okay, I have got some answers. Number B, number C, number D. So three different answers. Again, I have option received. Number D, number C, and then number B. Let's see what will be the correct answer. One second. Okay. Yeah. So the correct answer for problem number 6, it will be this number B, reactions which add a polar functional group to a drug. It is known, it is the closest description of the phase 1 metabolism. So basically metabolisms are divided into two phases. Phase 1 functionalization reactions and phase 2 are the conjugation reactions. Okay. So in the phase 1 metabolism, it converts the parent drug to a more polar metabolite by introducing and unmasking a functional group like OH, NH2 and SH, and phase 2 subsequent reaction in which a covalent linkage is formed between the functional group of the parent compound or phase 1 metabolite and an endogenous substrate such as the glucuronic acid, sulfate acetate or a amino acid. So phase 1 and phase 2 reactions, phase 1 reactions often make the drug sufficiently polar that can be excreted. However, phase 2 reactions are often required whereby a conjugate is added or covalently linked such as the amino acid, glucuronic, sulfuric or acetic acid to make the drug even more polar, charged or hydrophilic. So conjugation often occurs at the functional group site. So phase 1 reaction, the parent drug, phase 1 reaction, we have a functional group attachment more often. This functional group is the polar one, for example OH, NH2 and SH. Okay, and then phase 2 reactions, the parent drug, with the functional group, the conjugation happens. Although phase 1 reactions usually occur before phase 2 reactions, sometimes the order is reversed. The phase 2 reactions can occur alone also. Okay. So phase 1 reactions, more polar metabolite, that is pharmacologically uncharged, inactive, enhanced or otherwise modified. This phase 1 metabolites can go to the excretion or elimination, or phase 1 metabolites can go to the phase 2 reactions and have into the in the additions. Okay. So parent drug can also directly go to the conjugation or phase 2 reactions and all the metabolites will be eliminated subsequently. Okay. And this is the in the case of the metabolism, the most important thing that you have to notice that the importance of the metabolism is overall to make the lipophilic drug hydrophilic. Okay. So that it can be excreted. So this is the very important. As the metabolism goes on, the lipophilic drugs is getting more hydrophilic. Okay. The correct answer was number B, where it says the reaction which add a polar functional group to a drug. It is the closest description for the phase 1 metabolism. Okay. Now let me go to the next problem. Problem number 7, it says which of the following conversions is most unlikely to happen in the presence of CYP4P450. Your options are number A, aldehyde, RCHO to RCH2OH, alcohol. Then we have RCH2CH3 to RCOCH3. Then RR-CHX to RCOR- and number D, ARCH2R to ARCHOHR. Which of the following conversions is most unlikely to happen in presence of cytochrome P450? Most unlikely to happen. Okay, notice this term, that which of the following conversions is most unlikely to happen in presence of cytochrome P450? I will wait again for 2 minutes, then I will check the chat box. Okay, let me know A, B, C or D. Which of the following will be the correct answer? Which of the following conversions is most unlikely to happen in presence of cytochrome P450? Number A, RCHO to RCH2OH. Number B, RCH to CH3 to RCOCH3. Number C, RR-CHX to RCOR- and number D, ARCH to R to ARCHOHR. Which of the following conversions is most unlikely to happen in presence of cytochrome P450? Let me check. Okay. I think I have received only one answer, which is saying that number A is the correct one. Hmm. Okay. Others also should answer. Even if your answer is wrong, it does not matter because there is no marking for answering the class. Okay. So it's fine. I have received one answer, which is saying number A is the correct one. Let me now check if it is correct or no. So the correct answer will be yes, it is the number A, aldehyde to alcohol. This conversion is very unlikely to happen with the cytochrome P450. Now let us understand why. So phase 1 transformations are mainly catalyzed by the cytochrome P450 enzymes. The enzymes that constitute the cytochrome P450 family are most important metabolic enzymes and are located in the liver cell. They are the hemoprotein, they containing heme and iron, and they catalyzed a reaction that spills molecular oxygen such that the one of the oxygen atoms is introduced into the drug and the other ends up into the water. As a result, they belong to a general class of the enzymes which are known as the monooxygenase. Okay, so drug, we have a drug in presence of oxygen, and NADPH is the here is the coenzyme. So cytochrome P450 enzymes, and there must be protons also. So this enzyme basically they split the molecular oxygen and one oxygen is incorporated into the drug. That is why they are known as the monooxygenases, right? And another oxygen of the molecular oxygen ended up with the water and NADPH is converted to NADP. So this is the reaction which is catalyzed by the cytochrome P450. Now let us look at all the oxidative reaction that are catalyzed by cytochrome P450 enzymes on a saturated carbon center. Okay. So here we have the normal alkene RCNH3 which is converted to the RCH2OH alcohol which is oxidized again converted to the aldehyde to acid. Okay, so this is the oxidation of the terminal methyl groups. Okay, RCH to CH3 which goes to RCH to CH2OH, RCH to CHO and RCH to COOH. So this is the oxidation of the terminal methyl groups. Then if we have this penultimate or carbon of the alkyl substitution, for example RCH to CH3, the term this oxidation of the penultimate CH2 means this CH2 will give you the secondary alcohol at first and then it will give you the ketone. Okay, and also oxidation of the most exposed or most activated region of a cycloalkyl ring, for example in this position, the 4 position, here oxidation can happen. So these are the oxidation of the exposed alkyl groups and exposed regions of the cycloalkyl rings. Now oxidation of the activated carbon centers next to the sp2 and sp3 carbon, for example here the CH3 is oxidized to CH2OH and COOH, and here this RCO CH2, so here is the center is this carbonyl carbon is the activated center, and the next this carbon it is oxidized at first with with the alcohol. Here also this is the sp2 carbon, next to this carbon it will be oxidized to this one, and we have the triple bond which is the sp carbon which is also activated. Now this is the carbon next to this sp center which is oxidized like this, and for the ArCH2OH this reaction is similar. Then the dealkylation of amines, ethers, thioether and oxidation of activated carbon which can also happen. Okay, so first is the RCH2NR2 which is the primary amine, right? So this is first, this OH group is added to the next to the heteroatom like this one, and then what happens, the dealkylation happen means this molecule splits into the two part and the amine and aldehyde they get detached from each other. Same happens with the ether also, which is converted to aldehyde and alcohol, and we have this thiol, so it comes with aldehyde to thiol. Okay, and then dehalogenation of alkyl halide, for example this RrCHX to RCOX to subsequently ketone, this D The halogenation of alkyl halide, it is also happens in the phase 1 metabolism by cytochrome P450 enzyme. So all the reactions, all the examples are very, very important. Okay, so try to practice. The examples are very easy. Just you have to identify the correct reactions. Okay, so all these examples. So for now, number A, as you can see, this is the conversion of aldehyde to alcohol, which is not possible because it is a reduction reaction, right? So oxidation reactions or addition of with the addition of one oxygen, so it can happen. For example, number B, C and number D, all the reactions can be catalyzed by cytochrome P450, but not the number A. Okay. Now I will go to the next problem. Problem number 8, it says that alkenes and aromatic groups can be metabolized to diols. Which enzyme is involved? Your options are number A, cytochrome P450, number B, epoxide hydrolase, number C, both of the above, and number D, none of the above. The alkenes and aromatic groups can be metabolized into diols. Which enzymes are involved in this process? Your options are number A, cytochrome P450 enzyme, number B, epoxide hydrolase, number C, both of the above, and number D, none of the above. Alkenes and aromatic groups can be metabolized into diol. Which enzymes are responsible? Which enzyme is or is responsible? Cytochrome P450, epoxide hydrolase, both or none? Again, I will wait for some time, then I will check the chat box, okay? Alkenes and aromatic groups can be metabolized to diols by which enzyme? So alkenes and aromatic groups can be metabolized to diol. Which enzymes are involved? Let me now check if there is any answer in chat box. Number C, number C, number C. So I have received, I think, all answers as number C. Let us check if it is the correct answer or not. So the correct answer will be yes, it is number C. Both of the enzymes can actually metabolize this alkene and aromatic groups to diol. So first let us check. Okay. So cytochrome P450 enzymes can catalyze the oxidation of unsaturated sp2 and sp carbon centers present in the alkene, alkyne and aromatic ring. Okay. In the case of the alkene, the reactive epoxide is formed, which is deactivated by the enzyme epoxide dehydrolase to form a diol. In some cases, epoxide may evade the enzyme. If it happen, it can also act as an alkylating agent and react with nucleophilic group present in the proteins and nucleic acids, leading to toxicity. The oxidation of an aromatic ring results in and similar reactive epoxide intermediate which can have several possible fate. It may undergo to rearrangement reaction involving a hydride transfer to form a phenol. Normally at the para position, alternatively it may be deactivated by epoxide hydrolase to form a diol and react with the glutathione S transferase to form a conjugate. If epoxide intermediate invades this enzyme, it may be act as an alkylating agent and prove toxic. Electron-rich aromatic rings are likely to be epoxidized more quickly than the electron withdrawing substituent. Okay, and this has consequences for the drug design. So this is your double bond, which is epoxide is formed first. This addition of oxygen is done by cytochrome P450. Okay, and then water, in presence of water and epoxide hydrolase, we have this diol. Similar reaction happens in case of the cyclic alkenes also. Okay, and in case of aromatic ring also, this diol formation can happen, or rearrangement reaction can happen with a hydride shift, which creates a phenol at the usually preferred para position. Okay. So this is the reactions which is catalyzed by the cytochrome P450 and the epoxide hydrolase together. Okay. Now let me go to the next problem. So problem number 9 says identify the incorrect statement. Your options are: number A, cytochrome P450 can convert amine to an oxide. Number B, cytochrome P450 cannot oxidize heteroatoms. Number C, cytochrome P450 can convert thiol to disulfide. And number D, cytochrome P450 can perform desulfurization. Which of the following statements is incorrect? Number A, cytochrome P450 can convert amine to an oxide. Number B, cytochrome P450 cannot oxidize heteroatoms. Number C, cytochrome P450 can convert thiol to disulfide. Number D, cytochrome P450 can perform desulfurization. Which of the following statements is incorrect? Let me wait again for 2 minutes. Identify which of the following statement is incorrect. Cytochrome P450 can convert amine to an oxide. Cytochrome P450 cannot oxidize heteroatom. Cytochrome P450 can convert thiol to disulfide. Or number D, cytochrome P450 can perform desulfurization. What will be the correct answer? Which of the following statement is incorrect? Let me know. Let me check the chat box. Let's see if I have any answer. Okay, number B, number A, number D, number C, D, A, and B. So I have received 4 different options as answer. Okay, so let us check. So the statement which is incorrect will be, it is number B. Okay, CYP450 cannot oxidize heteroatom. So this statement is incorrect. I think I had number D as the answer in the chat box, right? Sorry, it is the number B. Okay, yeah, we have number B as the answer in the chat box. Very good. Now let us discuss about this particular question. So the okay, sorry. Yeah. So basically, I'll just look at the reactions. So all these oxidation reactions are performed by cytochrome P450. So tertiary amines are oxidized to N oxide as long as the alkyl groups are not sterically demanding. Okay. Primary and secondary amines are also oxidized to N oxide, but they are rapidly converted to hydroxylamine and beyond. Aromatic primary amines are oxidized to stage of the aromatic nitro group, a process which is related to the toxicity of the aromatic amines as the highly electrophilic intermediates are formed which can alkylate protein and nucleic acid. Aromatic primary amines can also be methylated into the phase 2 reaction to a secondary amine which can undergo phase 1 oxidation to produce formaldehyde and primary hydroxylamine. Primary and secondary amides can be oxidized to hydroxyl amides. Okay, these functional groups can also be linked to toxicity and carcinogenicity. Thiols can be oxidized to disulfide, and there is evidence that thiols can be methylated to methyl sulfide, which can be oxidized to sulfide and sulfones. Now, these are the tertiary amine. Okay, here an oxide formation can happen. Okay, secondary amine also the primary aliphatic primary amines can also become the this hydroxylation followed by the formation of the nitro intermediates. Okay, and then the primary aromatic and aromatic primary amines can also form the this aryl nitroarenes. Then we have RCH2NH, the primary amide. We can also form the hydrox this primary amide to that form this hydroxyl amide. Okay, this is the RCONHOH, and this is the aromatic ring, which is the pyridine kind of ring, can become inoxide in presence of this particular enzyme. Then we have the thiols, which can convert it to the disulfide. Okay, and CS, this desulfurization can also happen where the C double bond ends group 2 is converted to the C double bond O group. This is the oxidation of sulfur containing functional groups. Okay, thiol can be go to the oxidized to the methyl sulfide and then sulfoxide to sulfone, and also the phosphorus containing functional groups can be also oxidized with the help of this particular enzyme. So the statement number B, which says CYP 450 cannot oxidize heteroatom, this is false. As you can see, there are plenty of examples where CYP 450 can oxidize the heteroatoms, right? Now let me go to the next problem. Problem number 10 says indinavir is metabolized by number A glutathione transferase, number B CYP3A, number C epoxide hydrolase, and number D CYP2D9. Indinavir is metabolized by which of the following? Number A glutathione transferase, number B CYP3A, number C epoxide hydrolase, and number D CYP2D9. Which of the following will be the correct answer for problem number 10? Indinavir is metabolized by which of the following? Glutathione transferase, CYP3A, CY epoxide hydrolase, or CYP2D9. I will wait for a minute, then I will check the chat box. Let me know A, B, C, or D, which of the following will be the correct answer. So let me check if there is a correct answer for problem number 10, or is there any answer? Number A, number A, number B, number B. Okay, number B and number A. So I have received two different options, number A and number B. Now let me check which of the following will be the correct answer for problem number 10. So the correct answer will be, it is number B. CYP3A is the enzyme which is actually metabolizes this indinavir drug. Okay. So indinavir basically it is an antiviral drug Okay, so it is an antiviral drug used in the treatment of HIV and prone to metabolism, resulting in seven different metabolites. Okay, so indinavir, it gives rise to seven different metabolites. Okay, so the studies have shown that the CYP3A subfamily of cytochrome P450 family is responsible for six of these intermediate metabolites. Okay, so six of the metabolites, so among the seven, is caused by the cytochrome P3A subfamily of cytochrome P450 enzyme. The metabolites are concerned arise from the N-dealkylation of the piperazine ring. Okay, and this inoxidation of the pyridine ring, parahydroxylation of phenyl ring, and hydroxylation of indene ring. Okay, so here as you can see, here we have this piperazine ring. So the in dealkylation of the piperazine ring, in this position it gives rise to this metabolite, and then we have this parahydroxylation of phenyl ring. There is a phenyl ring there where the parahydroxylation happens. Okay, then we have the inoxidate formation of this pyridine ring, also the conjugation reaction. Okay, this is pyridine inoxidate formation of pyridine ring. Then we have this hydroxylation of indene ring, this hydroxylation of this indene ring, which after gives the N dealkylation and inoxidation. Okay, so these are the 1 2 3 4 5, and then these are the reaction. This is 1 2, and this 3, and then 4 5 6 and 7. So these are the, so these some six metabolites can be formed. So yeah, here 1, this is 2, this is 3, this is 4, this is 5, and this is the sixth number. Okay, so these all are formed with the cytochrome P450. Now the seventh metabolite is the glucocoordinate conjugate of the pyridine ring. This is the glucocoordinate formation conjugation of the pyridine ring. Okay, so this is the seventh metabolite. All the reactions occur individually to produce five separate metabolites. The remaining two metabolites arise from two of the metabolic reaction taking place in the same molecule. Okay, the major metabolites from this resulting from the dealkylation. As a result, the research has been carried out to try the design indinavir analogs such as a way to resist this metabolism. For example, structure having two methyl group in this position. If we have two methyl group in this position next to the okay, in this position, sorry, where is it? Huh. The metabolites research try to enable analogs research. For example, structures having two methyl groups on the activated carbon next to pyridine ring on this position. Okay, they have an effective on blocking the dealkylation. So as you can see, so in this position dealkylation happens. So if you can block this position, so this particular metabolism will not be done. Okay, so this is the now this is the analog of indinavir, which is resistant to the N dealkylation, because they are these methyl groups, they block this dealkylation of the piperazine ring. Okay, so this is the metabolism of indinavir, which is caused majorly by CYP3A subfamily. Okay, now let me go to the next problem. Problem number 11, it says which of the following enzyme is involved in the phase 1 metabolism. Which of the following enzymes is involved in the phase 1 metabolism? Your options are number A, flavin monooxygenase, number B, aldehyde dehydrogenase, number C, monoamine oxidase, and number D, all of these. Which of the following enzymes is involved in phase 1 metabolism? Number A, flavin monooxygenase, number B, aldehyde dehydrogenase, number C, monoamine oxidase, and number D, all of these. Which of the following enzymes is involved in the phase 1 metabolism? Let me know. A, B, C, or D. Which of the following will be the correct answer for problem number 11? Which of the following enzyme is involved in the phase 1 metabolism? Flavin monooxygenase, aldehyde dehydrogenase, monoamine oxidase, or all of these? I will wait for a minute, then I will check the chat box. Okay, let me know. A, B, C, or D. Which of the following will be the correct answer? Which of the following enzymes is involved in the phase 1 metabolism? Let me check the chat box. Okay, number T, number A, number T. So I have received three answers and majorly two different options, number A and number D. Okay, let me check which of the following will be the correct answer. So the correct answer will be yes, number D. All of these enzymes are involved in the phase 1 metabolism. Okay. So phase 1 reactions, these are so as you can see, we know phase 1 metabolism is the formation of the more polar conjugates from the starting material or the from the drug. Okay, so these are the phase 1 reaction that is catalyzed by flavine monooxygenase. So monooxygenase, as the name suggests, it will add one oxygen atom to the it will add one oxygen atom, or it will perform the oxidation reaction. Okay, so so for example, as you can see, for a formation of N oxide from RNH2, then the formation of this so this is the oxidation, addition of oxygen is the oxidation reaction. Okay, and so all these reactions are the oxidation reactions, and the phase 1 reactions which is catalyzed by the flavine monooxygenase. Then we have this alcohol dehydrogenase, where this alcohol, from primary alcohol it forms this aldehyde. Okay, and then from the secondary alcohol it forms the ketone. So alcohol dehydrogenases also are the phase 1 oxidative reactions examples. Then the example is the aldehyde dehydrogenase, which convert aldehyde to acid, and we have the monoamine oxidases also, which converts the primary amines to aldehydes. Okay, so all these are the example of the phase 1 oxidative reactions. Okay, so these are enzymes: flavine monooxygenase, aldehyde dehydrogenase, monoamine oxidase, and also alcohol dehydrogenase. All are the examples of the phase 1 oxidative reactions. Cause these enzymes take part in the phase 1 metabolism. Okay. So remembering each and every example is not important. Just this is the basic thing which you have to remember, that in case of the phase 1 metabolism, what is important, that the oxidation reaction will happen or more polar product will be formed from the starting material. Okay. Now let me go to the next problem. Problem number 12 says selegiline is a specific inhibitor of, your options are number A myo B, number B myo A, number C acetyl this ACHE, and number D ACE. Selegiline is a specific inhibitor of which enzyme? Let me know A, B, C or D, which of the following will be the correct answer for problem number 12. Again, I will wait for a minute or two. Then I will check the chat box. Okay, after 2 minutes, I will check the chat box. So let me know A, B, C or D, which of the following is the correct answer. Selegiline is a specific inhibitor of which of the following? Selegiline is a specific inhibitor of which of the following? Myo A, B, Myo A, acetylcholine esterase, or ACE, which is the angiotensin converting enzyme. Selegiline is a specific inhibitor of which of the following enzymes? Let me check the chat box. Okay. Number A, I have received only one answer, which is saying that number A is the correct answer. Okay, let me check if it is correct or not. So the correct answer will be yes, it is number A, myo B. Selegiline is a specific inhibitor of myo B. Okay. So designing the drugs to be isoenzyme selective. So this is the structure of the selegiline. Okay, as you can see, this is the structure. So designing drugs to be isoenzyme selective means that they can be designed to act on different diseases despite acting on the same enzyme. This is because isoenzymes differ in substrate specificity and are distributed differently in the body. Monoamine oxidase or MAO is one of the enzymes responsible for metabolism or the important neurotransmitters such as the dopamine, noradrenaline, serotonin, and exist in two isoforms, MyoA and MyoB. The isozymes differ in the substrate specificity, tissue distribution, and primary structure, but carry out the same reaction on the same mechanism. For example, this is the mechanism. This is the primary amine. The monoamine oxidase or MYO, then flavin coenzyme, they convert this to aldehyde, right? So MyoA is selective for noradrenaline and the serotonin, whereas MyoB is selective for the dopamine. This is important. The selectivity of MyoA, MyoB, although they carry out the same reaction, there is a selectivity. MyoA is selective for the noradrenaline and serotonin, whereas the MyoB, it is selective for the dopamine. Okay. MyoA inhibitor, for example, clorgelin, these are used as a clinically as the antidepressant. This is the MyoA inhibitor, clorgelin. This is used as antidepressant, whereas MyoB inhibitors such as the selegiline is administered with levodopa for the treatment of Parkinson disease. Okay, so levodopa and selegiline both are act together for the Parkinson disease. MyoB inhibition protects levodopa from metabolism. Clorgelin and selegiline are thought to act as a suicide substrates for but they are converted to by the enzyme to a reactive species that can react with the enzyme to form a covalent bond. The amine and alkene functional groups are present and the both drugs is crucial in this process. So as you can see, there is a amine. Okay, and there is a triple bond or alkyne functional group. These groups are important, and that's why then these groups make these particular drugs as a suicide substrate, where they convert the enzyme to a reactive species and form the covalent bond. Okay. So selegiline is all about the MyoA inhibitors. So yeah, selegiline is a specific inhibitor of MyoB. Now let me go to the next problem. Problem number 13, it says that phase 2 metabolism consists of dash reactions. Your options are number A hydrogenation, number B reduction, number C conjugation, and number D cleavage. Phase 2 metabolism consists of which reactions? Number A hydrogenation, number B reduction, number C conjugation, and number D cleavage. Again, I will wait for some time, then I will check the chat box. Let me know A, B, C, or D, which of the following will be the correct answer. Phase 2 metabolism consists of dash reactions: hydrogenation, reduction, conjugation, or cleavage. Phase 2 metabolism consists of which reactions? Hydrogenation, reduction, conjugation, or cleavage. Another minute I will wait, then I will check the chat box. So now let me check the chat box. Okay, number C, number C, number C. So I have received three answers and all are saying that number C is the correct answer. Let me check if it is correct or not. So phase 2 metabolism consists of which reactions? Yes, it is number C, conjugation reaction. Okay. So the most phase two reactions are the conjugation reaction catalyzed by the transferase enzymes. Okay. Now this is important to remember that this phase two conjugation reactions are catalyzed by the transferase enzymes. Okay. Yeah. The resulting conjugate are usually inactive, but there are exceptions to this rule also. So, for example, so now let us see what are the different phase two transformation. So, for first example is the glucuronation of the alcohol, phenol, and the carboxylic acid by the glucuronyl transferase. Okay, so this is the UDFH glucocorticoid. So, and this phenol, alcohol, and carboxylic acid, they can from this kind of conjugate, which is the beta D glucuronate conjugate. So here you can see there is a ring, there is an inversion of this anomeric center of the sugar, which happens during these conjugation reactions. Then we have this so similar glucocoordination of the miscellaneous functional group can happen, like for example with amide, with hydroxylamine, with sulfonamide, okay, amine, tertiary amine, thiol, with the primary amine, with the this secondary amine. This is not very much common, but it happens. Okay, so these are all the conjugation reaction which happens in presence of this UDFH glucocoordinate, which converts this particular this starting materials to the conjugates of the glucocoordinate. Then we have the formation of the amino acid conjugates. Okay, for example, there is a drug which has a carboxylic acid group. So ATP and acyl-CoA transferase converts this OH, attach this AMP function here, and then this thiol, which here it means this is CoA and coenzyme A and thiol, and then we have amino acid transferase and amino acid N acyl transferase, which transfers the amino acid, and this is the formation of the mechanism of formation of the amino acid conjugates. Okay, then we have this formation of the glutathione and mercuric acid conjugate from the alkyl halide. So from the alkyl halide RBR, we have this is the glutathione. This is the structure of glutathione, and glutathione transferase it converts this particular aldehyde alkyl halide to the glutathione conjugate, which is this one. And peptidases, then we have this peptidases, which basically what does this peptidases do? They basically cleave this peptide rings, and then the formation of the carboxylic acid and amines happen here. This carboxylic acid is converted to amine, and then acylation happens, and the acylation it can form the mercapturic acid conjugate, which is this one. Okay, this is the formation of the glutathione or mercapturic acid conjugates from the alkyl halide. And the last we have the formation of the glutathione conjugate, glutamate, cysteine, and glycine with the electrophilic group. So as you can see, this is the epoxide. Epoxide can form this glutathione conjugate. Okay, this is the normal alkene, which is also can form the glutathione conjugate. This is the epoxide, and then we have this sulfone type of SO2 type of molecule, which can also form the glutathione conjugate. So all these are the formation of the glutathione conjugate, which the electrophilic group. Okay, so what is the glutathione? As you can see, it is the glutamate, cysteine, and glycine together. It is known as the glutathione. So all this reaction, it sometimes it may little just feel a little complicated, but the majority you have to remember these functional groups here, and then what is the structure of glutathione. Okay, otherwise all the conversions are the similar, only the conjugation reactions happen, the functional groups they are converted to the particular conjugates. Okay, so all these are the examples of the phase 2 phase 2 transformations, which are the conjugation reaction, and it will be better if you can remember these transformations. Okay, if it is not possible to remember the structure of the glutathione, then it is fine, but try to remember the transformation, how they happen, the step by step. Okay, now I will go to the next problem. Problem number 14, which says that sulfation is an important method for the detoxification of the xenobiotics. Which one of the following acts as a source of the sulfate? Your options are number A, hydrogen sulfide, number B, methionine, number C, sulfuric acid, and number D, 3' phosphoadenosine phosphosulfate. Sulfation is an important method for the detoxification of xenobiotics. Which of the following acts as a source of the sulfate? Hydrogen sulfide, methionine, sulfuric acid, and phosphoadenosine phosphosulfate. Let me know A, B, C, or D. Which of the following will be the correct answer for problem number 14? Which of the following acts as a source of sulfate during the sulfation reaction? I will wait for a minute, then I will check the chat box. Let me know A, B, C, or D. Which of the following will be the correct answer? Sulfation is an important method for detoxification of xenobiotics. Which of the following acts as a source of sulfur? Hydrogen sulfide, methionine, sulfuric acid, or phosphoadenosine phosphosulfate? Let me check the chat box now. Okay, number D, number C. So I have received, I think, three different answers. One is saying number C, another is number D, and then number B. Okay, let me check which of the following will be the correct answer. So the correct answer will be number D. This is 3' phosphoadenosine 5' phosphosulfate is the source of sulfur during the sulfation reaction. So this is the structure, okay, of the phosphoadenosine phosphosulfate. This is the adenine ring is present here, and okay, yeah. So another form of conjugation is the sulfate conjugation. This is the less common than the glucuronation, and this restricted mainly to phenol, alcohol, arylamine, and N-hydroxy compound. The reaction is catalyzed by the sulfotransferase using cofactor 3' phosphoadenosine 5' phosphosulfate as the sulfate source. Okay. Primary and secondary amine, secondary alcohol, phenol form the stable conjugate, whereas the primary alcohol from the reactive sulfides, which can be act as a toxic alkylating agent. Aromatic hydroxylamine and hydroxylamide can also form the unstable sulfur conjugate, which can be toxic. And so there are we have phenol and alcohol. So the enzyme which is so there are two different things for this sulfur conjugation reactions or the sulfur conjugation. This is also known as sulfur conjugation phase 2 reaction or sulfation reaction. Okay, so in this case, two things are very important. One is the enzyme. Which enzyme it acts? It is the as here is it is mentioned that sulfotransferases are the enzyme which are responsible for these reactions, and the cofactor is 3' phosphoadenosine 5' phosphosulfate, which act as a source of the sulfur or source of the sulfate. Okay, so now alcohol and phenol with the in presence of this cofactor and this enzyme, it is converted to this sulfate conjugate. Like here we have the sulfate conjugate, and the amines, they are also converted to the sulfate conjugate. Okay, aromatic hydroxylamine and hydroxylamines can also form stable, they can form the stable sulfate conjugate that can be toxic. Okay, so this is the thing. So this structure, if it is not, if it is possible, try to remember. It is not very much important. So you can skip the structure also part, but remember the reactions which it catalyzes and how the reaction takes place. Okay. Now I will go to the next problem. Problem number 15. It says that which of the following statements is incorrect regarding the blood-brain barrier? Your options are: number A, the walls of the capillaries supplying blood to the brain are such which makes it difficult for a polar drugs to leave the capillaries. Number B, The capillaries in the brain have a fatty containing make it more difficult for the polar drugs to enter the brain. Number C, the inner walls of the capillaries supplying blood to the brain are hydrophobic in nature. And number D, hydrophilic drugs pass through the blood-brain barrier more easily than the hydrophobic drug. Which of the following statements regarding blood-brain barrier is incorrect? Again, I will repeat. Number A, the walls of the capillaries supplying blood to the brain are such which makes it difficult for polar drugs to leave the capillary. Number B, capillaries in the brain have a fatty coating making it more difficult for polar drugs to enter the brain. Number C, the inner walls of capillaries supplying blood to the brain are hydrophobic in nature. And number D, hydrophilic drugs pass through the blood-brain barrier more easily than the hydrophobic drugs. Which of the following statements regarding blood-brain barrier is incorrect or false? Read the options carefully and then let me know A, B, C or D which of the following you think is the correct answer for problem number 15. I will wait for a minute, then I will check the chat box. Which of the following statements is incorrect regarding blood-brain barrier? Number A, the walls of capillaries supplying blood to the brain are such which makes it difficult for a polar drugs to leave the capillaries. Number B, capillaries in the brain have a fatty coating making it more difficult for polar drugs to enter the brain. Number C, the inner walls of the capillaries supplying blood to the brain are hydrophobic in nature. Number D, hydrophilic drugs pass through the blood-brain barrier more easily than the hydrophobic drugs. I will wait for another minute, then I will check the chat box. Okay, let me know A, B, C, or D, which of the following you think is the correct answer. Let me now check the chat box. Okay, number B, number D. So I have received two different answers. One is saying number D, another is saying number B. Let me now check which of the following will be the correct answer or is there a different answer. So the correct answer will be number D. Okay, the statement which is incorrect, hydrophilic drugs pass through the blood-brain barrier more easily than hydrophobic. This statement is incorrect. Others, all the three statements are correct. So, the blood-brain barrier is an important barrier that the drugs have to negotiate if they want about to enter the brain. The blood capillaries feeding the blood to the brain are lined and tightly fitting cells which do not contain any pores, unlike the capillaries elsewhere in the body. Moreover, the capillaries are coated with a fatty layer formed by the nearby cells, providing an extra fatty barrier through which the drugs have to pass. Therefore, drugs entering the brain have to dissolve through the cell membrane of the capillaries and also through the fatty cells coating the capillaries. As a result, polar drugs such as penicillin do not enter the brain easily. The existence of the blood-brain barrier makes it possible to design drugs that will act at various parts of the body, for example, heart, and have no activity to brain, thus reducing any central nervous system or CNS side effect. This is done by increasing the polarity of the drug such that it does not cross the blood-brain barrier. However, drugs that are intended to act on the brain must be designed in such a way that they are able to cross the blood-brain barrier. This means they must have a minimum number of polar groups and have these groups marked temporarily by the product methodology. Having said that, some polar drugs can cross blood-brain barrier with the aid of the carrier proteins, while others like insulin can cross the process by pinocytosis described previously. The ability to cross the blood-brain barrier has an important bearing of the analgesic activity of opioid research that also been carried out to find ways of increasing the permeability of the blood-brain barrier using techniques such as the ultrasound and drugs such as the sildenafil. Okay. So this is all about the blood-brain barrier. So whatever drug we take for the for any problem of our body, like for the heart disease, for blood pressure, and so on, all the drugs they must not reach to our brain so that they cannot have the central nervous system side effects. So this is the blood-brain barrier. They actually protect our brain from other drugs which are not intended for the brain, but they are selective. So the design of the drugs which are intended to go to the brain or CNS, they must be like that. They can cross the blood-brain barrier. Okay. For example, they have to be highly hydrophobic, have a minimum polar groups, or if the polar groups are necessary for the active mechanism of action of the particular drug, it has to be masked by the prodrug therapy, and after reaching to the active site, that it can release the polar drug after crossing the blood-brain barrier. So these are the technique by which the drug which are intended for brain can be designed. Okay. So the statement which has said that the hydrophilic drugs can pass through the blood-brain barrier easily, it is not true. Other statement number A, which says the walls of the capillaries supply to blood to the brain are such that which makes it difficult for polar drugs to leave the capillaries, it is true. The capillaries of the brain have a extra fatty coating, making it more difficult for polar drugs to enter the brain. This is also true. The inner walls of capillaries supplying blood to the brain are hydrophobic in nature. This is also true. And number D, the statement is incorrect or false. Okay. Now let me go to the next problem. So problem number 16 says low concentration of the neurotransmitter dopamine has implicated in the Parkinson disease, and levodopa or L-dopa is the most widely used medicine to slow down the disease. Which of the following statement is incorrect? Your options are number A, dopamine cannot cross the blood-brain barrier. Number B, levodopa acts as a prodrug. Number C, levodopa can cross blood-brain barrier. And number D, levodopa first decarboxylated by dopamine decarboxylase before entering the brain. Which of the following statement is incorrect? Number A, dopamine cannot cross blood-brain barrier. Number D, levodopa acts as a prodrug. Number C, levodopa can cross blood-brain barrier. Number D, levodopa first decarboxylated by dopamine decarboxylase before entering to the brain. Which of the following statement regarding this levodopa is incorrect? Read the options carefully and then let me know. Which of the following statement is incorrect? Let me now check the chat box. Number B, number A, number D. Number D. So I have received two number D and one number A and one number B. Okay, let's see which of the following statement is incorrect. So the incorrect statement will be number D. Levodopa first decarboxylated by dopamine decarboxylase before entering the brain. This statement is incorrect. Okay, so basically levodopa, the amino acid levodopa is the biosynthetic precursor of dopamine. In the brain, levodopa is taken up by the dopaminergic terminal of the stratum and converted to dopamine by levoaromatic amino acid decarboxylase or decarboxylase. Dopamine is as such is it cannot use it because it does not enter the brain. Levodopa itself is a largely inert. Its effect depend on the increased synthesis of dopamine in the brain and are mostly related to the activation of D2 receptors. So this is the blood-brain barrier. So this is the levodopa. First, levodopa can cross the blood-brain barrier. Okay, and after crossing the blood-brain barrier, then it is decarboxylated. Okay, by this enzyme which is known as the dopa decarboxylate or levoaromatic acid decarboxylate enzyme to form the dopamine. So because basically this particular levodopa it acts as a prodrug. So dopamine it cannot cross the blood-brain barrier. That is why levodopa is designed, and this levodopa is most likely used as a prodrug and it can cross the blood-brain barrier. After the crossing blood-brain barrier, it is decarboxylated by dopamine decarboxylase. Okay, and so the statement number D, which says that levodopa is first decarboxylated by dopamine decarboxylase before entering the brain, this statement is false. After entering the brain, it is converted to the by this enzyme, not before entering the brain. Okay, and other three statements are true. Now I will go to the next problem. Problem number 17 says which of the following is not a primary pharmacokinetic parameter? Your options are number A, volume of distribution, number B, half-life, number C, clearance, and number D, all of the above. Which of the following is not a primary pharmacokinetic parameter? Volume of distribution, half-life, clearance, and or all of the above. Let me know A, B, C or D which of the following will be the correct answer for problem number 17. Which of the following is not a primary pharmacokinetic parameter? Volume of distribution, half-life, clearance, or all of the above? Which of the following is not a primary pharmacokinetic parameter? Volume of distribution, half-life, clearance, or all of the above? Let me check the chat box. Okay, I have received a couple of answers. Number B, number B, number B. Okay, I have received two answers. Both are saying that number B is the correct answer. Okay, let me check if it is so. So the correct answer will be yes, it is number B. Half-life is not a primary pharmacokinetic parameter. So basically, so first the drug is administered and when the drug reaches to the systematic circulation, after distribution the drug goes to the tissue and this is the equilibrium. So this is the known as the process of distribution, and this distribution is basically measured by the volume of distribution, this parameter. Okay, and then excretion and metabolism happen, and this metabolism and this process by which the drug in the systematic circulation, it is the amount of drug in the systematic circulation is decreases by these two process, excretion and metabolism. This is known as the clearance. Okay, so elimination is basically metabolism plus excretion, and disposition is a distribution plus elimination. So primary pharmacokinetic parameters are these: the clearance, volume of distribution. Now this primary pharmacokinetic parameters, and the secondary pharmacokinetic parameters are the half-life and bioavailability. Both this half-life and bioavailability, these depends on this volume of distribution and clearance. That's why they are the secondary pharmacokinetic parameter. Okay, so this is the volume of distribution curve. Okay, so this is the first is the amount of drug in the blood. So as when the drug is taken, then the absorption happens. So gradually, as you can see, the amount of drug in the blood it is increasing, and after that the amount decreases like this. Okay, and this is the Cmax, is the highest concentration of drug into the blood. This is the T half or half-life time at which the drug has lost half of its maximum concentration, and Cmin is the lowest concentration of the drug in the blood. that is this point. Okay, so this is this position, this complete overall drug exposure. It is the AUC or area under the curve. On the x-axis we have this time, and the y-axis we have the amount of drug. Okay, so this is very important. This is the pharmacokinetic. This graph depicts the whole pharmacokinetic. I mean, all the steps of pharmacokinetics very in a very big, good way. So these terms which you have to remember, this C max, T half, C min, volume of distribution, clearance, and so on. All this we will discuss one by one. Okay. So the terms we will be keeping, we will keep discussing in our upcoming slides, and also in upcoming class, in the next class also we will discuss about this particular term, because these are the very important, crucial regarding our eliminations also. Okay. So yeah, number B was the correct answer. Now, problem number 18. If a drug has a half-life of 4 hours, how much drug stays in our body after 24 hours? Your options are number A 1.56%, number B 50%, number C 6.25%, and number D 12.5%. If a drug has a half-life of 4 hours, how much drug stays in our body after 24 hours? 1.56%, 50%, 6.25%, or 12.5%? I will wait for 2 minutes, then I will check the chat box. Let me know A, B, C, or D, which of the following will be the correct answer. If a drug has a half-life of 4 hours, how much of the drug stays in the body after 24 hours? If a drug has a half-life of 4 hours, how much of the drug stays in our body after 24 hours? This is a very easy question. Let me check the chat box. I hope to find the answers there. Okay, number A, number C. So I have received two different options as answer, number A and number C. Let me check. So the correct answer will be, it is number A, 1.56. Okay. So half-life or T half of a drug is the time required for its concentration in the blood or total amount of the body to be in the blood, or the total amount in the body to be reduced by exactly 50%. Okay. So core principle, a 5 half-life rule. As a general rule, it takes approximately 4 to 5 half-lives for a drug to be considered effectively eliminated, clearing 94 to 97% of the substance. Steady state, when taking the drug regularly, it also takes about 5 half-lives to reach a steady state where the amount being taken is equal the amount being eliminated. Constant rate, for the most drugs following first order kinetics, the half-life remains constant regardless of the dose concentration. Why it matters clinically? Dosing frequency. Drugs with short half-life like insulin, glucagon, 42 minutes require frequent dosing, while those having high half-life, for example Prozac, may be dosed once a daily or less. Withdrawal. Medication with the shorter half-lives is often more acute withdrawal symptom if it stopped abruptly because they exit the system rapidly. Toxicity risks. If a drug has a long half-life, it can accumulate to toxic level if its doses are given too close together. Factors, they affect the half-life. Half-life is not a fixed number for every person. It is a hybrid value determined by two main pharmacophoric factor. That is why half-life is a secondary pharmacophoric factor, not the primary one. So what are the two factors on which the half-life depends? Is the clearance, how fast the liver or kidneys remove the drug. Lower clearance due to the kidney and liver disease increases the half-life. Volume of distribution, or VD, how widely the drug spreads to the tissue. Drugs that hide in the fat like Valium have the larger volume of distribution than the long and a longer half-life. So calculation formula, T half equal to 0.693 into VD divided by CL. This is the calculation formula for half-life. So now according to our question, the half-life has 4 hours. So after 4 hours we will have 50%. Then after 8 hours we will have the half of that, also 25%. After 12 hours we will have half of that, 12.5. After 16 hours we will have 6.25. After 20 hours we will have the half of this, 3.13. And then after 24 hours we will have 1.56% left. Okay, so the correct answer was number A, 1.56%. Then I will go to the next problem, problem number 19, which says dash is the study of the genetic variations between the individuals and the effect that has on the individual responses of drugs. Fill in the blanks. Your options are number A, pharmacokinetics, number B, pharmacodynamics, number C, pharmacomechanics, and number D, pharmacogenomics. Which of the following is the study of the genetic variations between individuals and the effect that has on individual responses of the drug? Pharmacokinetics, pharmacodynamics, pharmacogenomics, or pharmacomechanics, or pharmacogenomics. Let me know A, B, C, or D. Which of the following is the correct answer for problem number 19? Which of the following is the study of the genetic variations between individuals and the effect that has on the individual responses of drug? Pharmacokinetic, pharmacodynamics, pharmacomechanics, or pharmacogenomics? Let me check the chat box. Okay, number D, number A, number D. So I have three different answers, three answers I have received, and two are of which are saying number D and one of which is saying number A is the correct answer. Now let me check which of the following will be the correct answer. The correct answer will be number D, pharmacogenomics. Okay. So pharmacogenomics or PGX is the study of how an individual's unique genetic makeup affects their body's response to medications. By combining pharmacology, the study of drugs, and genomics, the study of genes, this field aims to move away from the one-size-fits-all approach to medicine toward the precision medicine, where the treatment is tailored for maximum efficiency and minimal side effects. How it works? Your DNA provides the instruction for building the protein, including enzymes that break down the drugs and receptors that drugs must attach in order to work. Genetic variations can result in number one, poor metabolizers. The body breaks down drug too slowly, causing it to build up in a toxic level. Ultra rapid metabolizers. The body processing a drug too quickly, meaning a standard dose may have a little to no effect. And then we have the target resistance. Genetic changes in the disease like tumor can make it resistant to a certain targeted therapies. Common application, pharmacogenomic testing is currently used for a growing number of conditions and medicines. Cardiology testing for cytochrome CYP2C19 variants, helping doctors to determine if the blood thinner clopidogrel will be effective. Oncology testing HER2 receptors determine if the breast cancer patient will benefit from trastuzumab, which is then psychiatry analysis of CYP2D6 CYP6 and CYP2C19 genes can guide the dosing of the antidepressant like the amitriptyline to avoid the adverse reaction. Infectious disease screening for the HLA-B5701 allele standard before prescribing HIV drug abacavir is prevent fatal hypersensitivity. So these are the common applications for the pharmacogenomics. Now the last question of today's session. Drugs can be termed as hard or soft based on dash. Fill in the blanks. Your options are number A excretion rate, number B bioavailability, number C metabolic susceptibility, and number D half-life. Drugs can be termed as hard or soft based on dash. You have to fill in the blanks. Number A excretion rate, number B bioavailability, number C metabolic susceptibility, and number D half-life. Drugs can be so what will be the correct answer for problem number 20? Let me wait for a minute or two, then I will check the chat box. Okay, this is the last question of today's session. Think carefully and then let me know A, B, C or D, which of the following will be the correct answer. Drugs can be termed as hard or soft based on which of the following: excretion rate, bioavailability, metabolic susceptibility, or half-life. Let me check. Okay, number C, number C. So I have received two answers and both are saying that number C is the correct answer. Okay, let me check if number C is the correct answer for the last question or not. So the correct answer will be yes, number C, metabolic susceptibility is the reason why the drugs can be termed as a hard and soft. So drugs, uh, so here, so drugs can be defined as a hard or soft with respect to their metabolic susceptibility. In this context, hard drugs are those that are resistant to metabolism and remain unchanged into the body, and soft drugs are designed to have a predictable controlled metabolism where they are inactivated to non-toxic metabolites and excreted. A group is normally incorporated which is susceptible to metabolism but will ensure that the drug survives for the sufficiently long period to achieve what is meant to do before the metabolized and excreted. Okay, drugs such that these are called the anti-drugs. Okay, so this is important. What is the anti-drug? Let me highlight this part. Okay. Yeah. So soft drugs are designed to have a predictable control mechanism which is inactivated to non-toxic metabolite and excreted. A group is normally incorporated into the soft drugs which is susceptible to the metabolism but will ensure that the drug survives for a certainly long time to achieve this which meant to before metabolism excreted. Drug of such of these are known as the anti-drugs. So the selection of synthesis of the hard and soft drugs. Drugs are divided into two types based on their metabolic susceptibility. Hard drugs. These can be defined as drugs that are biologically active and non-metabolizable in vivo, for example, enalapril, lisinopril, cromolyn, and bisphosphonates. And soft drugs, these can be defined as drugs that are produce produce predictable, controllable in vivo metabolism to form non-toxic product after they have shown in their therapeutic role. For example, cetylpyridine chlorides and soft chloramine. Okay. So yes, number C was the correct answer. So this was the last question of today's session. So today what we have discussed, mainly we have discussed about the metabolism, the phase 1, phase 2 metabolism. There is a huge number of reactions which takes place during the phase 1 and phase 2 metabolism, especially in the phase 1 metabolism, the polar reactions, also the conjugation reactions of phase 2 metabolism. Also we have discussed a little bit about the blood-brain barrier, its mechanism, and also we have discussed the various mechanism by which this metabolism or this things takes place. So this was our week 6 live session. Okay. I have already, I think, shown you where to find the materials. If you have joined late, also let me first share my email ID. So if you have any doubt or any query, please feel free to let me know. Okay, via this email ID. So any doubts, any query, please let me know via this email ID. Also, you can post your doubt or query to the discussion forum of the medicinal chemistry. So I'm portal medicinal chemistry. Where to find this problem solving session recording? On the first, you have to go to the announcement page of the NPTEL SWAYAM page of this medicinal chemistry. Here on the left panel, you will find there is a problem solving session January 2026. Clicking on that, you will go to the problem solving session recording, and if you click on that part, you will be redirected to a page which looks like this. It will contain all the YouTube links for the each and every class with a particular date, and also you will find the presentation folder link, which is the this is the Google Drive link which contains all the PPTs. Okay, so here you can find. So today it was our week 6 session where we discussed about this ADME, this drug metabolism and pharmacokinetics. In our next class, in the week 7, we will discuss the drug administration majorly, and then we will discuss about lead, the find or ways to administration routes to finding the lead, and also the product, the little bit we will discuss about, I think, in the next class. So that will be our upcoming Monday. Okay. So if you have any doubt or any query, please let me know. Otherwise, I will close the session for today. The presentation and class recording, it will be available in that particular page by tonight or by tomorrow morning. Okay, I will try to upload it as soon as possible. So you can check it if you have missed anything due to the poor network or anything, or you can let me know. If you have any doubt or any query from any previous classes, also you can let me know via email. So I will discuss that particular topic in the upcoming classes. Okay. Whatever we have done till now, whatever the topics we have covered till now, if you have any problem or any doubt, please feel free to reach me and let me know. Okay. If there is no question, I will close the session. Thank you for joining. Thank you for answering very well. Let's meet on the next Monday from 6 p.m. Okay. I will close the session now. Thank you.